Hello, everyone. Wait another couple of seconds just to make sure everyone is here. Yeah, got a couple more people joining us. Looking pretty bleak out there. Okay, so we might have a couple more people join us a little bit in, but looks like that's us for right now. Okay, so today we are going to pick back up with our discussion of the abdomen. Ooh, a lot of stuff on that bar. Hold on. That's what I wanted. Okay, so we are going to continue discussing the abdomen, and primarily today we're going to be discussing the different parts of the intestine. Okay, so here we have our, okay, go ahead and mute y'all. Okay, cool. So here are our objectives for today, mainly to do with our small and our large intestine. Okay. So when we left off, we had discussed how we have our esophagus coming in, we have our stomach, and we had left off essentially at our pylorus, which is where we're beginning today. Okay, so as we begin with our small intestine, we're going to be looking at three pieces: the duodenum, the jejunum, and the ileum. Okay, so your small intestine is going to go from your pylorus all the way to your ileocecal valve, which is where it will join the large intestine, also known as your colon. Now, the purpose of the small intestine is mainly to absorb ingested materials, so to absorb nutrients from the food you eat. So the way it's going to do this is that there are going to be these structures projecting out from the walls of the small intestine called intestinal villi, and they're going to be secreting digestive enzymes and helping your small intestine to absorb all the nutrients from the food you eat. Now, depending where we are in the small intestine, we're going to be absorbing and digesting different stuff, basically. So carbohydrates and protein are going to be digested in the duodenum and in the jejunum. The jejunum is also going to absorb fats. And then as we move down into the ileum, this is where we're going to be absorbing things like vitamin B12, bile salts, which are something that we partially used to absorb the fats in the jejunum, and then any other digestion products that we haven't absorbed. So we should have digested pretty much everything we need by the time we get to the end of the small intestine. All three sections are also going to be absorbing water and electrolytes. Throughout our small intestine, but really primarily located in the ileum, we're going to have some lymphoid tissue, so part of your immune system, basically, and structures called Peyer's patches. And you can imagine why that would be the case, right? In your GI system, you are eating food, you're eating things from the external environment. So it's important that we have basically like a trigger system for our immune system here as well. Okay. So first, let's talk about the duodenum. So this is the first part. So here we can see the stomach where we left off, our pylorus, which was the end of the stomach where we have our pyloric sphincter and our pyloric orifice. Okay. And this is leading us into the duodenum. So we're squeezing out chyme, which is our mixed up, chewed up food, and gastric juices. Okay. Now the duodenum as a whole is pretty short. It happens to be the widest part of your small intestine. And it's kind of C-shaped, essentially. Now parts of it are going to be mobile and intraperitoneal, but most of the duodenum is going to be held tight to the posterior abdominal wall. So we're going to think of parts of the duodenum as retroperitoneal because of the way they've been sort of plastered back to the posterior wall of the abdomen. Okay, so we are beginning at the pylorus. Okay, we have this C shape. Okay, now this is going to be surrounding the head of our pancreas, which we'll see later, but our pancreas, right, is going to be snuggled in there. It's where we'll see the head of the pancreas. And then they're going to end, or the duodenum is going to end, I should say, at the duodenojejunal flexure, which is on the left side. Now, this is located about 2 to 3 centimeters left of midline, and we're about the level of L2 at this point. So we are going to talk about the duodenum in parts, okay, because there's a lot going on when we think about the duodenum. Okay, so we have divided here our different parts of the duodenum by color. So we're going to have a superior part, we're going to have a descending part, an inferior part, and then an ascending part before we hit that duodenojejunal junction. Okay, and here we can see that pancreas snuggled in there inside our C shape. Okay. All right, so beginning with the first part, this is really short, about 5 centimeters. We consider the first 2 centimeters to be the ampulla of the duodenum, also sometimes referred to as the duodenal cap. It's special because it has a mesentery, which means it projects out into the space in the abdomen. It's intraperitoneal, and it's going to be mobile. So it's going to be able to move around a little bit because of that positioning. Okay. Now, the next 3 centimeters after that first 2 are not going to have a mesentery. They're going to be immobile. They're going to be plastered into the posterior wall of the abdomen. So we're going to consider them retroperitoneal. And at that point, they're kind of stuck, right? So as you're looking for the duodenum, you may be able to find this flexible end of it, that ampulla, those first 2 centimeters, but then it's going to be essentially diving back into the wall of the abdomen. Now, when we're looking at the superior portion of the duodenum, so this first segment, we're going to notice that it's actually overlapped by the liver and the gallbladder. So the liver and the gallbladder are going to be located over here. So they're going to be kind of covering it over. Now, the anterior portion of the duodenum is covered by peritoneum, but except for the ampulla, we don't have peritoneum covering the posterior part, which makes sense. It's only the ampulla that's really intraperitoneal there at the beginning, right? The rest of it is sucked back into the wall, retroperitoneal. Okay. When we're looking at the superior portion of the duodenum, we will also notice a ligament kind of fixing it into place. That'll be your hepatoduodenal ligament. So hepato for liver, duodenal for duodenum. Okay, so it's going to be attaching these 2 structures together, the liver and the duodenum. So we'll have this superior attachment to the liver, essentially, and it's going to be attached to our greater omentum inferiorly, which was that apron-like structure covering over our abdomen. Okay. So now we're going to move further down, all right? So we talked about this superior portion, okay? Now we're going to move to the second part, the descending portion, and then the third part and the fourth part are there. Okay, so the second portion of the duodenum is descending, flowing down, right? If we're tracking food and now chyme through it, it's going to be longer than that first superior part. Okay. It's roughly descending along the sides of the L1 to L3 vertebrae. So those would be your skeletal landmarks for this. Okay. And this is where we have that C-shaped curve, and we could find the head of the pancreas. Now, the whole second part of the duodenum is retroperitoneal. Okay. And what we're also going to see in this area is we're going to see something called the hepatopancreatic ampulla, or the ampulla of Vater. This is going to be where we have our bile duct and our main pancreatic duct entering into the duodenum and secreting essentially digestive enzymes in order to help us digest food. Okay. We're going to refer to the space essentially where this ampulla is opening as the major duodenal papilla. So that's just going to be kind of our name for this area where we have the opening. The more common name is the one in parentheses, just so you know, this ampulla of Vater. Okay. Now, as we go across this flexure, right, we have a duodenal, inferior duodenal flexure. A flexure just means a turn. we're going to enter into this third part of the duodenum going across the L3 vertebra, essentially. So this is going to lie pretty much horizontally. Okay. Nothing specific additional we need to know about that yet. Okay. But what we are going to notice about the fourth part, as we are ascending and sending ourselves over and into the jejunum, we're going to have an interesting structure called the ligament of Treitz. So this is a slip of skeletal muscle actually coming from the diaphragm that we kind of hook around. Okay. So you have a slip of skeletal muscle. And this is going to suspend that flexure between the duodenum and the jejunum. So that transition point is going to be marked by this ligament, essentially, right? And this also is a really important landmark in surgery for finding where this transition point is. And it has some functional implications too for movement of digestive products through the small intestine. So taking a look at that ligament, so it's connecting posterior to the duodenojejunal flexure, and it's going all the way back posterior behind the pancreas. And when it contracts, because of its movement, it's going to help move contents of the duodenum forward, essentially. So we can see here that sharp flexure between the duodenum and the jejunum. So that's our, that should be a J. The duodenum and the jejunum. And there we see that suspensory ligament that is actually made up of skeletal muscle a little bit. So it can contract and kind of move stuff around a little for us. All right. So in terms of vasculature, the arterial blood flow is going to be coming from the celiac trunk. So that's that first little branch that we see off the aorta, right? So we have our aorta running here. Right, so celiac trunk, okay, is going to be supplying the duodenum, but we are also going to be having a little bit of blood flow from our next branch down, our superior mesenteric artery. Okay, so also a little bit from here. So from the celiac trunk, we have our gastroduodenal artery and our superior pancreaticoduodenal artery that are branching off of our hepatic artery. So our first branch is here going to the liver, the hepatic artery, and then we have these branches down, supplying both duodenum and a little bit to the pancreas. Okay. And then when we're talking about these third and fourth parts of the duodenum, we have a branch from this SMA, that's superior mesenteric artery, wherever you see that. All right, just because it's a long word. Okay. So from the SMA, we also have an inferior pancreaticoduodenal artery. So you can see that going to the duodenum and to the pancreas. Okay. So we have a sort of mixing of blood flow here at this point. We have a little bit of anastomosis. So the duodenum is going to be considered an important kind of transition point when we're thinking about blood supply to the digestive tract, because we are having this transition between blood going from the celiac trunk to blood from the superior mesenteric artery. This is because the duodenum is essentially a transition between what we'd consider the foregut and the midgut if we were looking at the embryological origin of our gut structures. In terms of the venous drainage of the duodenum, they're essentially going to follow the arteries. What we're going to talk about later on in this lecture is that when we talk about the veins draining the gut, all of these branches are going to have the same names, right? So we are going to have similarly named veins to our arteries, like the superior mesenteric vein, like the superior mesenteric artery, right? We're going to talk about these, though, because they are actually going to be following back, many of them, to something called the hepatic portal vein, which is going to be slightly different than what we have happening with our arteries, because this means that much of the blood going through our abdomen is going to have to actually go back through the liver for processing before it gets back into your circulation. And we'll look at a map at that in a bit. Okay. In terms of lymphatics, your anterior portion, your anterior lymphatic drainage is going to go to the pancreaticoduodenal lymph nodes, which lie along the arteries, the pancreaticoduodenal arteries. And then they're going to travel back to the pyloric lymph nodes, which lie along the gastroduodenal artery. So essentially, we're going kind of back towards our stomach is what we see happening here. All right, so going back this way. In terms of our posterior lymphatic drainage, this is where we're going to go back towards the superior mesenteric lymph nodes. Okay, so this is going a little bit different in terms of our direction. Okay, so we can see those there. Okay. In terms of innervation, our innervation is coming from the vagus nerve when we're thinking about our parasympathetics, so our rest and digest. And sympathetic innervation is coming from the greater and lesser splanchnic nerves. So after our duodenojejunal flexure, we are in the jejunum. So that's going to be our second part of the small intestine. Okay. So here in this image, our jejunum is everything in blue. Okay. Our duodenum is in red, like the actual red. This pinker stuff is colon. Okay. And our ileum, the third part of our small intestine, is in yellow. Okay. So the jejunum is going to also be intraperitoneal. Okay. And it's mostly going to be in the left upper quadrant. Okay. So most of it is up here in the left upper quadrant, although we can see that there are bits of it that are projecting into other portions. So unlike the division between the duodenum and the jejunum, there isn't actually like a really clear line between the jejunum and the ileum. So we are going to want to be able to identify some of the characteristics of each, especially if you're trying to like resect a specific portion of the small intestine. But you're not going to be looking for any specific landmark. Unfortunately, there's not like a clear ligament to use as a dividing line. So the next table is going to have some comparisons, some things that tend to differ, but this is going to be a little bit of a gradation. One of the things, though, we can see is that position, not an exact dividing line, right? But we can see ileum in this right lower quadrant, bit in the left lower quadrant, most of the jejunum up here in the left upper quadrant. So when we're looking at the jejunum and the ileum, we can see some differences in the coloration a little bit. We can see differences in the thickness of the wall. Some of the vascular properties are going to vary. There are a number of different things we can look at to kind of try and tell these two apart. So you can look through those later. This is a good reference slide if you're trying to figure that out. So moving down into that ileum, this is the third part of the small intestine, mostly in the right lower quadrant, but as you can see, it occupies a lot of area, so it's not a strict hard and fast rule. But what is helpful is that the terminal part of the ileum, so its last part where it ends, is usually going to be basically practically in the pelvis as we enter into the cecum. So this is really the bottom corner of our abdomen here that we're going to find. Okay. So that's going to be the end of our ileum. Okay. Before we move on to what's going on as we enter the cecum, we want to take a look at the mesentery. Okay, so the mesentery of the jejunum and the ileum is really important because we know it's carrying blood flow both to the small bowel and then away from it, as well as containing those lymph nodes. So we know that the mesenteries in general are basically two layers of peritoneum sandwiched against each other, filled with fat. They look actually pretty wrinkly. So we have an actual image, it's kind of blurry here from surgery though. We can see those kind of wrinkles. It's containing all of these important structures within it. So it looks like it's just a big lump of fat, but it's actually very important. It's kind of fan-shaped, actually, when you see it in real life. Those wrinkles are going to make it look kind of like a zigzag kind of accordion fan kind of structure. Now, it actually has a specific configuration and a specific place where it attaches. So if you do take small intestine out, resect it, and put it back, you're going to want to put it back in the correct orientation so that all those blood vessels stay oriented correctly, don't get cut off, and so that you maintain appropriate blood flow to the small bowel. You don't want to put it back in such a way that you're compressing arterial flow or venous flow and then end up with the tissue dying. So for this reason, you do want to pay attention to the root of the mesentery, because that's going to help you put it back in the right orientation. So the root of the mesentery, so its attachment to the rest of the body, is going to be directed obliquely, so at an angle, inferiorly, and to the right. Okay, so when you're in lab, you can practice taking a look at that. Okay, so running through it, we have the superior mesenteric vessels, lymph nodes, fat, as well as the autonomic nerves going to the small bowel. So there's a lot of important stuff going through here. So we'll take a look here at the vasculature. Okay, so when we're looking at vasculature, this is going to be coming from the superior mesenteric artery. Okay, so that second prong coming off of the aorta, essentially, which is then going to give off a jejunal artery and an ileal artery, which anastomose with each other. Okay, so the superior mesenteric artery, that branch, the initial branch off the aorta, is coming off at about L1, about a centimeter inferior to the celiac trunk. So if you're tracking down on the aorta, you'll see it quite soon after the celiac artery. It then enters into the mesentery and is splitting out. So we can talk about some of the structures of branching that we see here in the mesentery itself. So we do have our jejunal and ileal arteries, okay, right? But we can also see that we have all these loops of structure and then these straight ends coming off of them, okay? So these loops and arches that we're looking at, these are called our arterial arcades, okay? So these arches are our arcades, okay? And then the straight bits are called the vasa recta, okay? So branching off of each of these loops, we have straight portions, okay? Going out to the small bowel, those are the vasa recta here. Now, in terms of venous drainage, we are going to have a superior mesenteric vein matching that superior mesenteric artery that is going to drain both the jejunum and the ileum, and it's going to go back into the hepatic portal vein. So we're going back through the liver, actually. This is going to be anterior and to the right of the superior mesenteric artery if you're looking for it in the body. So in terms of lymphatics, we saw on that cartoon image some little green dots. That's because we do have lymphatic vessels and lymph nodes here in the mesentery, okay? And we are going to have them in a couple different places, and we want to pay attention a little bit to the flow here. So the juxta-intestinal lymph nodes are going to be just along the edges of the intestinal wall, essentially. So juxta-intestinal, next to the intestines. Okay. Now, along those arcades that we were just looking at, those loops, you're going to have mesenteric lymph nodes. Okay. So we can see some of those here. Okay. And these are draining in order back up to your superior central lymph nodes, which are going to lie along the artery, along the superior mesenteric artery. Okay. So this is how we are draining our lymph back up and in. Okay. The one difference is that when you're looking at the terminal part of the ileum, we do have a specialized structure. So you do have ileocolic lymph nodes here when we're talking about that specific kind of juncture there. All right. In terms of innervation, we're again going to have for our sympathetic innervation some splanchnic nerves. So we have the lesser splanchnic nerves coming through plexuses, so the celiac plexus and the superior mesenteric plexus. Your sympathetic nervous system is that fight or flight activity. So when your sympathetic nerves are more active, this is going to reduce anything to do with digestion. So you're going to reduce secretion of digestive enzymes. You're going to reduce peristaltic contractions that usually move fluid along, and you're going to divert blood flow away from the intestine and probably towards like skeletal muscle in the heart. For parasympathetic innervation, it's doing the opposite. Parasympathetic innervation to the jejunum and the ileum comes from the vagus nerve. So that wandering cranial nerve, wandering all the way down from the head, all the way down here to your intestines. Okay. So when your vagus nerve is activated, lots of parasympathetics to your gut. This is going to increase peristalsis, increase secretion of those digestive enzymes, and increase blood flow to the intestines so that you can do that rest and digest activity. Now, remember, the intestines are pretty insensitive to pain, right? We were talking about the difference between the ability of the visceral peritoneum and the parietal peritoneum to sense a lot of things, right? And we said that our parietal peritoneum can localize pain pretty decently, has the ability to sense pain, the ability to sense temperature, so on and so forth. But the intestines are covered in that visceral peritoneum, so they really can't sense all that much. What they can sense is distension. So what your intestines are able to sense distension, right? If they're swelling up and out, if they're getting wider, right? They can sense that distension, and it's going to feel like a cramp. So it produces colic, which is just another word for intestinal cramps. So we're next going to move into our colon, which is another word for the large intestine. So the duodenum, jejunum, and ileum together made up our small intestine. The ileum is going to lead us straight into the cecum, which is the beginning of our colon or our large intestine. Now, in the small intestine, we hopefully digested everything we're planning to digest. So what we're left with in the colon is anything that we are unable to digest. So we're really just moving fluid and solidified materials along here. And we're also going to be absorbing some more water as we move along. Okay, so we're going to be converting a more liquidy kind of waste into a semi-solid waste as we move through the colon. We're also going to be storing waste products temporarily here in the colon until defecation, right? So in order, our portions are going to be the cecum. So we're going to begin with the cecum here, which has a little appendage, the appendix, coming off of it. We're then going to move into the ascending colon, going up, go across our transverse colon, and then go down our descending colon, our sigmoid colon. This is kind of S shape, leading us into our rectum, and then ultimately into our anal canal. Okay. Those are going to be our portions of our colon or our large intestine. All right. Now, if we're trying to tell the difference, because we're in the abdomen, we want to tell the difference between our colon and our small intestine. Super important so that we can orient ourselves to where we are, that we have the right thing that we're looking at. There are a number of ways to do this. So first time you see them, they may look pretty similar, but when you actually concentrate, they're not too bad, not too hard to tell apart. So one thing we're going to notice is that the colon, our large intestine, has something called omental appendices or epiploic appendages on it. So these are going to look just like little extra globs of fat kind of hanging off of the large intestine. You won't see those on the small intestine. So that's a really big giveaway. Another thing you see on the large intestine that you don't on the small intestine is this kind of line down the center. Okay. So we have this line. This is actually longitudinal bands of smooth muscle. Okay. And so when it contracts, it's going to be able to shorten the colon, which is going to create a sort of bagging area, right? We're going to create these little bags as we contract this, right? And it wrinkles, okay? And so you'll actually see the demarcations of these little segments, these little bags. Those are called the haustra, okay? So the name of this longitudinal line is the Taenia coli. And the haustra are these little sacs that are formed when it contracts as it sort of folds in. Okay. It is also true that the large intestine is larger than the small intestine. When we say larger, what we really mean is wider. We're talking about the caliber of the intestine, so like its diameter. So it will be wider than your small intestine. So let's talk about the different parts. Okay. So we had our duodenum, our jejunum, our ileum. Okay. And now we're at the final part of the ileum. Okay, so we have our ileocecal junction here where we're joining up between our ileum and our cecum. Okay, so the cecum is going to be the first part of the colon, and it's not going to have a super obvious division necessarily from the ascending colon. They're continuous with each other. So we're going to continue to go up as we move our digestive products along. Now, the cecum can be found pretty easily. So it lies in the iliac fossa in the right lower quadrant. And if it's distended and full of feces, you can actually palpate it through the abdominal wall. So the anterolateral abdominal wall, potentially, there's a lot of stuff in the cecum. You could be able to find this just by pressing on it. Okay. So it is almost entirely covered by peritoneum, so that layer, but it doesn't have a mesentery, so it's not going to have that same sort of flexibility as our small intestine did, and as some of the other parts of our colon will have. So it's not all that movable. It's really going to be in that location. Now, we do have what we refer to as a valve where the ileum is entering the cecum. So this is our ileocecal valve. They used to think that there was a sphincter here kind of controlling the release of our feces, I guess now, into the cecum. But as far as we can tell at this point, it's actually pretty passive. So it's really more just a flap kind of preventing reflux from the cecum back into the ileum. Okay. We would also refer to this as the ileal papilla, if we're referring to this center. So the valve, if we're referring to the whole structure, the papilla, if we're referring to like the opening, basically. For our arterial supply here, we have our ileocolic artery. So the ileocolic artery is the last branch of our superior mesenteric artery, the terminal branch. Venous drainage is going to go through our superior mesenteric vein. Lymphatic drainage is also going through an ileocolic lymph node this time and going up to our superior mesenteric lymph nodes. Innervation is coming from the superior mesenteric plexus when we're thinking about sympathetics, still vagus if we're thinking about parasympathetics. So we remember the words ileocolic and superior mesenteric. When we're thinking about the cecum, we should be good to go. Now, we do want to pay attention to the appendix, primarily because of appendicitis, because when it gets infected and inflamed, it's very painful. If it were to burst, that would be very potentially dangerous because you'd have a lot of things that should not be collecting in your intraperitoneal space, escaping out of your large intestine. Okay, so technically we think of the appendix as a diverticulum, so an extension essentially of the colon that is blind, so it doesn't end. It's a dead end essentially. We currently don't think that the appendix has any actual real function in people today, although every now and again there are articles coming out about possible functions of the appendix, but we basically think it's a vestige of evolution. It's about 6 to 10 centimeters in length. This will be variable, contains some lymphoid tissue, and you're going to see it in a variety of potential positions. So it does have a mesentery, so it's going to be flexible, it's going to be mobile. And it can be in a number of different positions. The most common is a retrocecal position. Okay, so here we see retrocecal in that dotted line, so like behind the cecum. But these are all potential options of the appendix. This is not someone with like a giant star-shaped appendix. This is just showing you different places we might see the appendix positioned. And this will make a difference to patients presenting symptoms, because you're going to be feeling pain primarily from tissue that's getting irritated, rather than localizing it from the appendix itself necessarily. Okay. So in terms of arterial supply, there's a branch to the appendix from the ileocolic artery. So we have an artery we just call the appendicular artery. And your venous drainage is ultimately going back to the superior mesenteric vein. So it's kind of following along with what was going on for our cecum. So the lymphatic drainage is going back to the ileocolic lymph nodes as well, and then back to the superior mesenteric lymph nodes. All right, so that's going to lead us to the ascending colon. So we had our cecum, we had the appendix hanging off of it. Now we're moving up and eventually out. Okay, so the ascending colon is the part of the colon that extends from the cecum to the liver. Okay, so our cecum was here, so we're thinking like here-ish, up towards this corner, which is commonly called the hepatic flexure in actual practice, although books like to use terms just referencing the anatomy. So you will also see right colic flexure in your book, but hepatic is more common, partially because then people don't have to think about right and left. You can just think, oh, it's next to the liver, it's the hepatic flexure. Okay. So the ascending colon is going to be a bit narrower than the cecum. So you'll notice the cecum will be more bulging. Then we'll see it narrow a little bit as we flow into the ascending colon. And we'll also see that the ascending colon is retroperitoneal. So it's going to be stuck to the posterior abdominal wall. It is covered in peritoneum anteriorly and on its sides. And actually in about a quarter of people, it does kind of have a mesentery, so it might project a bit intraperitoneally, but because of its strong attachment to that posterior wall, we really would still consider it retroperitoneal overall. We won't really change the definition just for that anatomical variation. Okay. Next to it, we do have a gutter where we can have fluid drain. So we would have our right pericolic gutter right next to it. Okay. And in terms of its supply, it's going to be getting arterial blood flow from branches of that ileocolic artery, as well as the right colic artery. Okay. This is coming off the superior mesenteric artery, joining right next to the right middle colic artery in the arcades. There is also a marginal artery that kind of runs along the length of the colon near its border, that kind of gets incorporated into the arcades, those loops running around. Okay. But primarily, remember, ileocolic and right colic artery. Okay. Venous drainage, ileocolic and right colic veins to the superior mesenteric vein. And then lymphatic drainage is going to go to epicolic or pericolic lymph nodes, going back to the ileocolic and right colic lymph nodes, so matching that arterial and venous supply, and then back to your superior mesenteric lymph nodes. Innervation is coming from your superior mesenteric nerve plexus. All right, so we're still thinking that SMA here, essentially. So next, we're going to move into the transverse colon. So the transverse colon is actually the longest portion of your colon, and it's the most mobile. So it's going to be really movable. We are going to see that it has a mesentery called the transverse mesocolon. So the position of that transverse colon can actually vary quite widely. So you may see this in a lot of different regions, depending how full it is, depending on a lot of different things. So you might see it as high as L3, you might see it almost down in the pelvis, okay? What you will notice, regardless, is it's going to have a hepatic or right colic flexure. So we have by the liver, hepatic, a turning point, right? So as the ascending colon turns into the transverse colon, we have to cross a corner. So that's that hepatic flexure. And then we'll have another corner at its end, which is your splenic flexure or your left colic flexure. And you're going to see that that's higher up, right? Okay, so one of these turns is going to be higher than the other. Okay, so that splenic flexure is more superior. It also has a sharper angle, right? Sharper angle than the hepatic flexure did. When you look at it relative to other structures, you'll see that it's kind of anterior to the inferior pole of our left kidney. It's also going to be attached to the diaphragm. So we have a phrenicocholic ligament attaching, the diaphragm to our colon. Okay, so phrenicocholic ligament is partially creating that splenic flexure. Now, when we think about the transverse mesocolon, that mesentery suspending the transverse colon and allowing it all that mobility, we're going to see that it's adherent to the posterior wall of the omental bursa. So what we're saying is the omental bursa is related to that lesser sac, that sort of space in the smaller space that we had in the abdomen. So we're going back through that space essentially to adhere to the posterior wall of the abdomen. And when we're looking at the root of the transverse mesocolon, we're going to see that it's running kind of along the inferior border of the pancreas as it runs into the parietal peritoneum. Okay. So for arterial supply, we are going to see that we have a middle colic artery coming from the superior mesenteric artery, supplying that transverse colon. And the marginal artery, that one that was kind of lying along the arcades around the colon, can also help supply the transverse colon. So we have that anastomosis there, basically. And our venous drainage is going to go to our superior mesenteric vein. Lymphatic drainage pretty much matches. So our middle colic lymph nodes to our superior mesenteric lymph nodes. And innervation is still coming from that superior mesenteric nerve plexus. All right. So we're now going to move into the descending colon. So we've gone through the cecum, the ascending colon, the transverse, the colon, across the splenic flexure. Now we're going back down through the descending colon. So like the ascending colon, the descending colon on the left side is going to be retroperitoneal. So we're going to have peritoneum lining covering its anterior portion and laterally, but its posterior portion is going to be really bound to that posterior abdominal wall. It'll have a little bit of a short mesentery, but it's not really entering into that intraperitoneal space. Okay, it's not really mobile. We're running from our splenic or left colic flexure down to our sigmoid colon, this S shape. Okay, and we have another gutter where we can have fluid flow lying along it. So we also have a left paracolic gutter. So that's our orientation here. Okay, and we don't have many specific things that we need to know about the descending colon before we lead into our sigmoid colon. Okay, so into our sigmoid colon, we're going to go into an S-shaped loop now. Okay, we got our S-shaped loop. Okay, variable in length a little bit, but we're connecting the descending colon to the rectum, kind of going down through that bowl of the pelvis. So that sigmoid colon is going to lie from the iliac fossa, then down to S3, so that third vertebra in the sacrum, where we're going to join into the rectum. We then have a junction between the rectum and the sigmoid colon called the rectosigmoid junction. This is where we're going to see the Taenia coli, that line that we saw down our large intestine, end essentially. So that's how we're going to kind of know from the outside that we have entered the rectum. That's going to be about 15 centimeters from the anus. And the sigmoid colon does have a long mesentery, so it's going to have a lot of range of motion when we're thinking about the sigmoid colon, which means that it is at a higher risk of basically twisting and getting messed up. So formally, we would say that the sigmoid colon has a higher risk of volvulus. So that was the term we used before for when an organ twists its orientation and potentially constricts like its own blood flow. So basically, the more range of motion you have for a structure, the more risk you have for something like that occurring. When we look at the sigmoid mesocolon itself, so that mesentery for the sigmoid colon, we're going to notice that it's actually kind of V-shaped. And it's going to run along the external iliac vessels back to the area of the sacrum. And part of the reason we want to note that shape is the right posterior to the apex of that V of the mesenteric going to the sigmoid colon. We're going to find our left ureter, as well as the division of the left common iliac artery. Now that's important because if you were dissecting the area of the sigmoid colon out in surgery, right, and you go a little too deep to that apex of the sigmoid mesocolon, potentially cut the ureter, that's a big problem. So we always want to be aware of where we are and what other things we need to be careful of. Okay. So the V, that shape of the sigmoid mesocolon, is also helping you find where your left ureter is. Okay. So we're going to look at the arterial and venous blood flow for that descending and sigmoid colon kind of together, as well as the lymphatic drainage and the innervation, which is why it wasn't on the slide for the descending colon separately. Cool. All right. So we are now moving down from before we had our superior mesenteric artery, right? And we were looking at all its branches, right? We had our ileocolic artery going here. We talked about our right colic artery. We had our middle colic artery and its branches, right? And that got us basically across the transverse mesocolon. Now that we are moving into the descending and the sigmoid colon, we want to think about our third little branch off the aorta going to the intestines and the contents of the abdomen, our inferior mesenteric artery. So if you're looking for these in lab, there's a much bigger space between the superior mesenteric artery and the inferior mesenteric artery than there was between the superior mesenteric artery and your celiac trunk, which is just like a centimeter. So you're looking a lot farther down when you're looking at that separation. So basically what we're looking at here is we're seeing a change in the blood supply to the digestive tract. So when we're thinking about our blood flow transition points, our celiac trunk, that first little bit, not marked on here, but it's more like here-ish, right? It's going to do our upper digestive tract, so like the stomach till the duodenum. Our superior mesenteric artery is doing everything from the duodenum all the way to our splenic flexure, essentially. And then the inferior mesenteric artery is supplying our splenic flexure on down, okay? So you do want to visualize the junction between the superior mesenteric artery and the inferior mesenteric artery when you're doing a colon resection, because we do have extensive anastomosis here, right? So we can see that this anastomosis, this connection, right, is going all the way around the colon, right? So that's important for our blood flow. And we want to make sure that we are preserving adequate blood flow to all of the parts of the colon instead of transecting that and potentially cutting off and killing a big part of our colon. So you want to be able to find that. Okay. For venous drainage, for these areas that are supplied by our inferior mesenteric artery, we also have an inferior mesenteric vein. Okay, so our blood flow is going to go along that matching vein. Our lymphatics are going to be named similarly to the lymphatics for our ascending colon. So we're going to have epicolic and paracolic lymph nodes leading to intermediate colic lymph nodes into the inferior mesenteric lymph nodes now, named for that inferior mesenteric artery instead of going back up to the superior mesenteric ones, like we saw on the right side. Okay. Slight difference because that splenic flexure is kind of our transition point. If you're looking right at the area of the splenic flexure, the lymph might be partially draining into those superior mesenteric lymph nodes because it's kind of close to that as well, right? So at the transition point, you're going to want to just be a little careful. Okay. So for innervation now, this is also where we're going to see a change in where we're getting innervation from. So now we're going to start to see our lumbar splenic nerves coming up and giving us our sympathetic innervation, so from the lumbar region. And now instead of our vagus nerve, we have our pelvic splenic nerves supplying our parasympathetics, okay, as we pass this transition point. We will talk a bit more about these structures as we talk about our pelvis and perineum, but I want to finish out our digestive tract. So as we're moving from our sigmoid colon, right, we saw that we had that rectosigmoid junction, right? So we have this joining point. We would have seen our tinea coli end here as we lead into the rectum. Now, these next structures are going to be retroperitoneal and subperitoneal, so like below the lining of the peritoneum, and we'll find them at the level of S3 on down. They are kind of the terminal part of the colon. These are structures that are allowing us to defecate, so to actually expel these waste products. We'll go into more detail on their structures as we talk about the pelvis. Okay. All right. So we are now going to talk about some of our kind of accessory organs, or other organs in the abdomen. So we've gone through the digestive tract. Today, we're also going to talk about the spleen, the liver, the gallbladder, and a little bit about the pancreas. Okay. So we're going to start with the spleen. Okay, so the spleen is ovoid in shape. It's kind of purple in color. It's about the size of your fist, although you will see that size change in certain diseases, essentially. It is your largest lymphatic organ in the body, and it has a lot of important roles. So it's going to be involved in lymphocyte production. It's going to be involved in immune surveillance and responding to that immune surveillance. It also helps you recycle blood cells and platelet components and can act as a reservoir for red blood cells and platelets. Now, its capsule is pretty delicate. So it has a fibroelastic capsule, which is great when we want to change blood flow to the spleen and sort of upregulate some of these important functions, allowing it to expand and contract. But that also makes it kind of vulnerable. So it's pretty soft, it's very vascular and sinusoidal. So it's going to be quite vulnerable to damage. In terms of our quadrants, we would say that the spleen is located in the left upper quadrant in its superolateral portion. And the spleen in its normal position is protected by essentially the 9th to 11th ribs from the posterior. So it shouldn't be coming down out of that costal margin under normal conditions. It's only really in pathologic states when the spleen is growing in size that you'll see it kind of peeking out below the ribs, essentially. So when it grows and expands, it's going to expand inferomedially, and that's why you might see it going below the costal margin then. It's going to rest on the splenic flexure of the colon, so you can see that association in the image, right? So we have our splenic flexure named for the spleen that's sitting right there on top of that splenic flexure. Now, you might think, given the list of pretty important functions, that this would be a vital organ. And by vital, we really mean required to sustain life. But it's not. We do have backup, essentially, for all of these four functions of the spleen. So it is important because it does these things, but you can survive without it. Other parts of your organ systems will take over some of these functions. Okay. Parts of the spleen. So we have this curved structure here. And on the opposite side, we have the splenic hilum. That's where we have our arteries and veins entering and exiting the spleen. Okay. Speaking of those arteries, they are quite large. Okay, so we can see them here, and we can see a couple other structures as well now. Okay, so we can see that we have a gastrosplenic ligament, a connector between the stomach and the spleen. The stomach is not actually pictured on here except for just this little part, but that gastrosplenic ligament would be kind of behind and between back there. Okay. There's also a connection to the kidney. So there is a splenorenal ligament as well. So here we see kidney. Okay, so there's a connection there as well. Okay. So looking at our arterial supply and venous drainage, so your splenic artery is actually the largest branch of your celiac trunk. So here we can see that celiac trunk right here. Okay. So this is our celiac trunk. And then you can see that splenic artery going off to the left, going to the spleen, going into the hilum. Okay. Venous drainage is going to come back through a splenic vein. The splenic vein is going to meet up with your inferior mesenteric vein and then continue in and meet up with the superior mesenteric vein posterior to the neck of the pancreas. Then they're going to lead together, probably, into the liver. And I say probably because when we think about veins, there are some common variations. Lymphatic drainage is going to be going into the pancreaticosplenic lymph nodes, which lead into the celiac lymph nodes. So basically going back to where that blood flow is coming from. Innervation, also coming from that celiac plexus, so a nerve plexus around that celiac trunk. Next thing up is our pancreas. So your pancreas is primarily, well, it also has multiple functions. When we're thinking about the abdomen, we largely think about the digestive function of the pancreas, but your pancreas has both exocrine and endocrine function. But all of this is kind of related to eating. So in terms of exocrine function, that refers to the pancreas's ability to create these digestive enzymes. Our acinar cells are going to secrete pancreatic juice full of these pancreatic enzymes that are going to go through the pancreatic duct. Okay, pancreatic duct, you can see here, right? And ultimately enter the duodenum along with the bile duct at the ampulla of Vater. Okay. Onto this little lumpy area that we're using to locate our duodenal papilla. The other big function of the pancreas that you may actually have thought more about, right, is its endocrine function. So it does produce two really important hormones. In these small islets of Langerhans, we have cells that produce glucagon and insulin, which enter the bloodstream and basically adjust our cells' response to glucose, essentially. So they shift us between absorptive and post-absorptive states. Okay, so exocrine function, those enzymes going through the pancreatic duct to the ampulla of Vater. Endocrine function, hormones, glucagon, and insulin coming from our islets of Langerhans. In terms of where we're going to find it, so it's going to be retroperitoneal, so it's going to be along the posterior wall of the abdomen. It's going to be crossing transversely at about L1 and L2, and you're going to be posterior to the stomach. We also noticed when we looked at the shape of the duodenum that it was C-shaped, right? And we mentioned that the head of the pancreas is snuggled right up in there, in that C shape of the duodenum. So we're in this image, we're posterior to the stomach for basically all of it. We see this head of the pancreas. And as we move out towards the tail of the pancreas, we'd be going up towards our spleen, pretty much. Okay, so you'd locate it transversely between the duodenum and spleen at about the level of L1, L2. So for arteries, we're going to have branches coming from the splenic artery as well as from the gastroduodenal artery. So our spleen was over here, right? Our stomach is lying anterior to this. This is our duodenum. So branches from the spleen, splenic artery, branches from the arterial flow to the stomach and duodenum are also going to the pancreas. For our veins, our venous drainage is going to go into pancreatic veins that drain into the splenic vein. Lymphatics, we have pancreaticosplenic lymph nodes, which go to the superior mesenteric or celiac lymph nodes via some hepatic lymph nodes. So there's kind of complicated lymphatics going on here. In terms of innervation, we are kind of still in this foregut, midgut kind of region. So we got our vagus nerve, and we also have some abdominopelvic splanchnic nerves involved here. Okay, so we're going to look a little closer at the parts of the pancreas. So when we look at the divisions, we are going to think of the pancreas as having a head, a little neck, a body, and a tail. Okay, so we're snuggled in this C shape of the duodenum with the head. We're reaching out with our tail towards the spleen. This is our spleen here. Okay, all right. So beginning with the head, we have an expanded kind of portion fitting into that C shape. It's actually sitting and resting on the inferior vena cava, as well as the renal arteries and veins. Now, also through this region, we have the bile duct. It's actually going to be kind of embedded in the pancreas as it's running through this area. And it's joining together with the pancreatic duct, which was yellow in the previous image, isn't on here, but they're coming together at that ampulla of Bader and going into the duodenum. So there's a lot of stuff going on here as we're looking at the head of the pancreas. The head of the pancreas is also considered to have a little additional process. So this portion, more or less, is the uncinate process. This is like a projection from the inferior part of the head of the pancreas. So it's got a little extra bit. The neck of the pancreas, we really just consider the portion right next to the pylorus of the stomach. So the neck would just be like a little narrow band, essentially. All right. Then the body is going to run along the aorta and along the L2 vertebra, anterior surface covered by peritoneum, posterior doesn't have peritoneum, it's in direct contact with the aorta, as well as with the superior mesenteric artery, renal vessels, the left adrenal gland, the left kidney. And this is essentially like lying in the floor of the omental bursa, so that lesser sac in the abdomen, forming part of the stomach bed, by which we really just mean the structures that the stomach is resting upon. Okay. And the tail, heading out to the spleen, is going to be located anterior to the left kidney. It's going to come real close to the splenic flexure and the spleniculum, and it's going to be kind of mobile. So it's going to be able to move back and forth a little bit as it passes between the splenorenal ligament and the splenic blood vessels. So we can see that kind of positioning on this cross-sectional image. So here we see our spleen. Here we see kidney, adrenal gland, and here's our pancreas kind of curving around. Okay, so we can imagine that has a little space to move around here. So next we're moving into the liver. So the liver is really your largest gland in your body, and we consider it the largest single organ after the skin. Its capsule is more fibrous than the capsule we had around the spleen, so it's a little more protected by the Glisson capsule. Except for the fats that we are absorbing, all the nutrients coming into your body from the GI tract are initially going to be kind of sent through the liver. So the liver kind of acts, among its many functions, as like a processing center for a lot of the things coming into your body. It also stores glycogen, so essentially stored glucose kind of on tap for your body. You got a good system for storage for that here in the liver. It also helps, well, it does produce bile, which is a yellow-brown to greenish fluid. It's produced continuously in the liver, helps you emulsify fats, so helps you break up the fats in order to digest fats. We're going to see that that bile is stored in the gallbladder. So it's produced by the liver, stored in the gallbladder, and then delivered to the duodenum via the bile ducts, one of which we've seen as we were thinking about the duodenum and the ampulla of Vater. So when we're thinking about the position of the liver, we're going to be in the right upper quadrant, deep to ribs 7 to 11. The liver does move, so it's sitting right under the diaphragm, basically. So as the diaphragm moves, the liver will move. So that's going to be important to remember as you're looking for it on exam, as you're learning to palpate it during exam. Now, the diaphragmatic surface of the liver is going to be convex and smooth. On the opposite side, on the visceral surface, so the part that's facing all your abdominal organs, it's going to be concave. It's going to have a bunch of fissures and impressions where you have other organs kind of coming into contact with it. There are two potential spaces between essentially the liver and the diaphragm. So we have our falciform ligament connecting our liver up to the walls of our abdomen. On either side, we have a subphrenic recess where you could potentially get collection of materials between the liver and the diaphragm. Okay. Now, we have another area we want to know about, something called Morison's pouch, or the hepatorenal recess. So this is a space that we see between the liver and the kidney. So it's an extension of the space below the liver, so an extension of the subhepatic space. And it's going to be between the right part of the liver and the right kidney and adrenal gland. So this is an area you might be looking at if you are examining someone after abdominal trauma. And really what you'd be looking for here is just whether you see blood collecting here. So a FAST or EFAST POCUS exam during trauma, you might be looking for blood collection right here between the liver and kidney in that Morison's pouch. Since it's white here, you don't have blood, this area would appear kind of black if we had fluid filling it. Other areas of the liver, so the liver is mostly covered with visceral peritoneum, but it's not covered with visceral peritoneum on its posterior surface. So it's just directly touching the diaphragm there. And there's also a deep groove on that bare area where your inferior vena cava is running. The liver also has a fossa for the gallbladder, where the gallbladder is going to snuggle into a little hollow of the liver. This area also is not covered by visceral peritoneum. An important region that we're going to find is something called the portal triad. So we're going to see the bile duct, the hepatic artery, and the hepatic portal vein all coming into essentially the same area when we look at the liver. So those three together, we're going to consider our portal triad. So now we're going to look at some diagrams to understand some of this surface anatomy and some of these relationships. Okay. So right now we're looking at the visceral surface of the liver. So we're looking at that kind of underside in contact with our abdominal organs. And we can see that it forms kind of like an H. Okay. So this H-like structure on the concave visceral surface has names. So these fissures are our umbilical fissure. So this is a continuous groove formed by the round ligament, which is a remnant of the umbilical vein. The porta hepatis is the part that we want to think of most importantly. So the porta hepatis is this transverse fissure where we're going to have that portal triad, the hepatic portal vein, the hepatic artery, hepatic ducts, also lymphatic nerves, hepatic nerve plexus, et cetera, coming in and also leaving. So that porta hepatis fissure is really important. And then we also have a right sagittal fissure. This is going to be a groove that's formed by the fossa for the gallbladder on the anterior portion, and then a groove for the vena cava on its posterior portion. Okay. So we got an H, porta hepatis is in the middle, right sagittal fissure, you'd see the gallbladder anteriorly, vena cava posteriorly, and then the left sagittal fissure is formed by remnants of the umbilical vein, which we call the round ligament of the liver here. All right. So this creates a series of lobes. So we can look at the liver here, both from superior and inferior. And I want to give you a heads up just that they're going to be both anatomical lobes of the liver as well as sort of resectable lobes of the liver later on, and they're not exactly the same. So when we're thinking anatomically, we think about two lobes just separated out by the falciform ligament as our starting point. Okay, so we're going to have a left lobe and a right lobe. Okay. And then if we were to turn the liver over, we'd have two accessory lobes that are basically part of the right lobe. So we have our caudate lobe and our quadrate lobe. Note that the quadrate lobe is right next to the gallbladder. Okay, so the caudate lobe isn't actually quite caudal in position, but it has a little tail, which is why they named it that way. Okay. So those are anatomical lobes of the liver. Okay. We're going to think about its blood supply. Okay, so the liver has a really interesting blood supply because it does get some of its blood for the tissue of the liver itself from the hepatic artery. So the hepatic artery proper, branching off your celiac trunk, it's supplying the non-parenchymal structures of the liver with arterial blood. So it's supplying like bile ducts and stuff. But the rest of the liver is getting its blood flow mainly from the hepatic portal vein. So that means we actually have the liver being supplied by partially deoxygenated blood, because that blood coming into the hepatic portal vein, right, is venous blood. However, it's still kind of convenient for us because it is carrying nutrients that we've absorbed from the small intestine. This means that we have the ability to modify those nutrients. We have the ability to detoxify things before they really enter the rest of the body, because we have that blood flow running through the liver, which has those functions. So like if you have a glass of wine, right, absorb that alcohol through the walls of your intestines, that alcohol is ultimately going to go into those veins, right? Lead you back to the hepatic portal vein, and it's going to go through the liver, which can then ideally detoxify that alcohol, right? Like neutralize that alcohol before it gets to the rest of your body, depending on how much you had, right? So that's kind of what the liver is doing in general, as relates to all sorts of different things that you might be ingesting. Okay. In terms of drainage of the liver itself, the liver does have hepatic veins. So there are going to be central veins in something called the hepatic lobule, forming collecting veins, combining to form hepatic veins, which ultimately get you back into the inferior vena cava. Okay. So we're taking venous blood flow from the gut, sending it through the hepatic portal vein into the liver for processing, detoxifying, neutralizing, modifying, but the blood does eventually get us back into systemic circulation, into the inferior vena cava. So we're kind of like a pit stop. Okay. So this is showing that, again, essentially just kind of written out now. Okay. So the hepatic portal vein is really important because it's moving blood from the GI tract and also from the spleen into the liver, which is then able to filter, process whatever is coming through in that venous blood. Okay. So the hepatic portal vein is usually formed by the union of the superior mesenteric and splenic veins. In about a third of people, the inferior mesenteric vein is also joining in. So exactly what these connections look like is going to be a little bit variable. Okay, but this is what we're talking about essentially. right? So we're going back to the hepatic portal vein to the liver here. Okay, cool. So the hepatic portal vein is large in terms of like diameter, but it's quite short. So it's going to be located within the hepatoduodenal ligament. So the ligament that's attaching your liver to your duodenum. It's going to reach the porta hepatis, which is that central part of the H, that fissure, where it's then going to divide into a right branch and a left branch. Okay. So it's called a portal system because a portal system is when we have like multiple capillary beds kind of in sequence in contact with each other. So the hepatic portal system is connecting capillaries of the intestines to modified capillaries in the liver. So those are the two capillary beds that are making it a quote unquote portal system. If people refer to a portal system, they are usually referring to this portal system, I think. Okay. But presumably clear from context. Okay. So the hepatic portal system is helping you filter whatever you're absorbing from your intestines, process these things, also helping you process some of the components that you're getting from the spleen as it breaks down red blood cells. Also going to help you cleanse blood of potentially bacteria that could get picked up by the blood as it's absorbing things from the intestines. Okay, so when you're really going to pay attention to the portal system is when there's something going wrong with it. So blood can basically get kind of like backed up into the portal system, especially when you have liver diseases, right? So imagine you have a disease of the liver, like cirrhosis or like a tumor, right? You are decently likely to make it just like more difficult for blood to flow through it, right? Which means that the blood that is trying to come back from the GI tract, get filtered by the liver, and ultimately come back to your systemic circulation is going to kind of like build up. So you can end up with an increase in blood pressure and a backup in that portal system. Okay. So this means that when you have someone with chronic liver disease, you're basically going to see a bunch of vascular indications of that. So potentially ascites, so buildup of fluid in the abdomen itself, esophageal varices, spider nevi, caput medusae, and palmar arrhythmia are all vascular findings that you might find that have to do with that buildup of blood pressure in the portal system. Okay. All right. So now we're going to talk about lobes of the liver from a functional perspective. Okay. So anatomically, we had the right, the left, the caudate, and the quadrate. But what's probably going to be more interesting to you clinically is that you actually have fully independent subdivisions of the liver, essentially. So the liver is continuous with itself, but you are going to have 8 surgically resectable hepatic segments where you can take one away and there's really no problem, which is why you can have living liver donors, right? You can give away part of your liver and be perfectly fine. Okay, so what we see on this image are those functional subdivisions of the liver. So you do still have a right lobe and a left lobe, but they are no longer quite the same. So up at the top, this is showing you, I believe, the anatomical divisions, right? So you can see that the right lobe anatomically was much bigger than the left lobe. But when we think about the right part of the liver in terms of these subdivisions versus the left part, they're much more equal, right? So the right and left here, okay? And then you can see that we divide multiple times until we get down to those 8 surgically resectable hepatic segments. And they're resectable, right? Like you can take them out independently because they each have an independent branch of the portal triad, right? So they have their own branch for arteries, veins, and for bile, essentially. Okay, so those are, I think, practically more important than the anatomical lobes, although of course this is an anatomy class, so don't ignore the anatomical lobes. Okay, so another function of the liver, other than just processing, it's really important, is its production of bile. So bile is made by the liver, stored and concentrated in the gallbladder. And by concentrated, we just really mean some water can get absorbed. And the gallbladder is then acting as a storage tank until you need bile in order to digest fats. So bile emulsifies fats, makes the little fat globules small enough that you can digest them. So it's like waiting for you to eat a big greasy cheeseburger or something. I don't know. So when you need that bile, your gallbladder is going to contract in response to a hormone, CCK. And at that point, it's going to expel that bile into the duodenum, that ampulla of Vater. Then you'll have that nice bile there, giving you nice bile salts that help you digest the fats. Okay. So we can see the structures that lead to this here. Okay, so we are going to have right and left hepatic ducts coming out of the right and left parts of the liver. Okay, cool. So they're coming here. All right. The porta hepatis is that little groove of the H if we're looking at the visceral surface of the liver. So that's the location we're in. Okay. And so pretty much right after that, they're going to join together to become the common hepatic duct. Okay. So we can see that here. And here we see the location of the gallbladder. Okay. So if we're storing our bile, we might loop around and go into it. But what we want to know for ourselves right now is just that there's this connection, right? So we have a common hepatic duct and we have a cystic duct with the gallbladder. And I'm pointing it out that way in that directionality because then they're going to form together into the common bile duct, which is actually what leads us into the duodenum. Okay. So the common bile duct is connected to both the cystic duct and the common hepatic duct. Okay. We're then going to have further joining. So that common bile duct is joining with the main pancreatic duct at the ampulla of Vater. So we're shooting all sorts of enzymes essentially into the duodenum there. There is a sphincter at the end of the common bile duct that kind of controls entry of the bile into the duodenum. So that sphincter is called the sphincter of Oddi. And here you can see an MRI image essentially. So in terms of the function of the gallbladder, it can hold up to 50 milliliters of bile and sitting on the visceral surface of the liver, snuggled in its own little fossa. And it is anterior to the superior part of the duodenum. So it's contracting in response to the hormone CCK. So that hormone CCK gets secreted by the duodenum when it senses fatty foods. That makes the gallbladder contract, sending bile out through the cystic duct, out through the common bile duct, and ultimately into the duodenum itself. The gallbladder has a fundus, a top part, a body, a main part, a neck, which is a narrow-shaped S portion that leads you into the cystic duct. The arterial supply here is the cystic artery, which comes from the right hepatic artery, venous drainage through the cystic vein. And then for lymphatics, we're going back up to the same lymph nodes that we're sharing with the liver, so to the hepatic lymph nodes. A thing we want to note here about our region with the gallbladder is that we have something called the triangle of Callot that we can use as kind of a landmark for finding some of these structures. So this is our cystohepatic triangle. So it's going to be formed by our common hepatic duct, our cystic duct, and then the surface of the liver itself. Okay. So we're kind of making a triangle like this. And we're doing this so that we can find the cystic artery. So the cystic artery is going to be somewhere within this triangle, but you can note that there is variation here in where we find exactly the position of that cystic artery. That's what this horrible diagram C is about, I think. I'm told there are probably even more variations than that, right? So regardless, you're going to be looking in this triangle for the cystic artery. And this is like, if you're trying to remove the gallbladder, important that you are aware of its blood flow. You want to clamp that down as well, right? So you're going to need to be able to find that blood flow first. So you're going to look for that triangle of Callot to find that cystic artery. Okay, so that's what we got for today, and I'll see you guys next week. enjoy your week, chugging along towards spring break.
Thank you.
Yep, take care.
Thank you.
Thank you.